everyone, welcome to my channel Pure Biology, and today I come with another video, that is the nuclear RNA polymerase. Okay, so in my previous video I have already explained about the eukaryotic transcription. So here I will try to explain that which factors are important for the eukaryotic transcription. That means eukaryotic transcription is a complex process and it is done by the different types of factors. So one of the important factor is the RNA polymerase. Okay. So here in this video we will learn about the RNA polymerase. How this RNA polymerase, or what is the role of the RNA polymerase in RNA transcription process. Okay. So here the eukaryotes, So eukaryotes have three different polymerases. One is the RNA Pol 1, second is the RNA Pol 2, and third is the RNA Pol 3. So RNA Pol 1, that is responsible for the synthesizing most of the rRNA, small r RNA. Okay, RNA Pol 2, that help in the synthesizing process of mRNA and most of the snRNA. So RNA Pol 2 types of RNA synthesis, that is dependent on the RNA Pol 2, that is mRNA and snRNA. And RNA Pol 3, that synthesizes a variety of the small stable RNAs, that including tRNA, 5S, small rRNA, U6 snRNA. Okay, so these are the role of the RNA polymerase 1, RNA polymerase 2 and RNA polymerase 3. Now here is thing that the eukaryotic RNA polymerase, these are the large proteins. That means these are made up of the large proteins, and each RNA pol is a multi-subunit protein. That means the large protein is made up of the different subunit proteins. Okay, and the eukaryotic Pol 2, that consists of the 12 subunits. Pol 2, that means RNA polymerase 2, that consists of 12 subunits, and the two largest subunits are the homologous to the bacterial beta and beta dash subunits. So here in case of generally all the RNA polymerase, these are made up of the large proteins and the protein that is made up of with the small subunits. But in case of particularly the Pol 2, it has 12 subunits and 2 large subunits. These are the homologous. That means these are same type of. And the two subunits, that this beta and the beta dash, also subunits that present in the bacterial RNA polymerase 2. In addition to the increased number of subunits, eukaryotic Pol 2 differs from the prokaryotic counterpart in that it has a series of heptad repeats with the consensus sequence and the terminal of the largest Pol 2 subunits. Okay, one thing is that subunit, this is the part. So what is the difference between, that means the RNA Pol 2 both present in the eukaryotes and the prokaryotes. So difference is what? That increasing number of subunits are present. That means in case of prokaryotes, the RNA Pol 2 consists of the number of subunits that increase in the eukaryotic Pol 2. Okay, so number of subunits are become increasing from prokaryotes to eukaryotes, and these are the main differences between the prokaryotic Pol 2 and the eukaryotic Pol 2. Clear? Now there is another thing is that a series of heptad repeats also present, and the sequence, that is TYR, ACR, PRO, THR, ACR, PRO and ACR. This sequence also be present at the carboxyl terminal of the largest Pol 2 subunit. So what is saying that in case of largest Pol 2 subunit, there is another thing present, that is the sequence, this consensus sequence, and heptad repeats. These are also present. Okay, and this carboxyl terminal domain is both a substrate for several kinases. Okay, another thing I have to first check. So difference is what that? Difference is that heptide repeats, consensus sequences, and the carboxyl terminal units or carboxyl terminal domain, you can say. This domain is attached with the largest pol to subunit. So these are the factors. These are not factors. These are the important differences by which we can easily identify the prokaryotic RNA Pol 2 and the eukaryotic RNA Pol 2. Clear? Now, what is this carboxyl terminal domain? This carboxyl terminal domain is that, okay, this is both a substrate for the several kinases and a binding site for a wide array of proteins. So actually, what is this carboxyl terminal domain? This is actually the site where the substrate can be bind, and also it is the site for binding of the proteins. Clear? So this is the actually CTD. Now what is saying that in case of plant genome, these are the Pol 2, Pol 3 that present in that E. coli. E. coli is one kind of eukaryote obviously, and also but in case of plant, you can say that in animal cell, if we consider, these three polymerases are very important. But in case of plant, also two polymerases are discovered, that Pol 4 and Pol 5. This is the Pol 4 and Pol 5. And these are evolved from actually Pol 2. Okay, so these Pol 4 and Pol 5 that are already identified in the plant genome, but these arise from where? These arise from RNA Pol 2. Now, RNA Pol 4, this is important for what? This is important for the biogenesis or the synthesis of siRNA, and Pol 5 that transcripts for RNA directed DNA methylation. Okay, so the role of this RNA Pol 4, that is the siRNA synthesis, and Pol 5, that is the DNA methylation. In case of mitochondria and the chloroplast, where we know already that their DNA is totally different from the nuclear DNA. Clear? So here in case of mitochondrial RNA Pol, so there is another RNA Pol that is not present in the or that is different from the nuclear RNA Pol. Clear? This RNA Pol is encoded by the nuclear genes, and it is heterodimer. This is the heterodimer, and this is composed of two subunits. There are two subunits. One subunit is related to the monomeric RNA Pol and of the bacteriophage T7, and the other resembles alpha factor of the eubacterial RNA Pol. So what is saying that in case of mitochondrial RNA Pol, there are two subunits. Okay, in mitochondrial RNA Pol, there are two subunits. One subunit that is totally just like monomeric RNA Pol of bacteriophage T7, just resemble to the monomeric RNA Pol of the bacteriophage T7, and the other subunit that is resembles to the alpha factor of the eubacterial RNA Pol. Clear? So these are the thing that is present in the mitochondria. But in case of the chloroplast of the higher plants, here the two types of RNA Pol present. The chloroplast encoded RNA Pol, this is one thing, and nuclear encoded RNA Pol. One is chloroplast encoded RNA Pol, In the case of chloroplast, there are two types of RNA Pol. One is the chloroplast encoded RNA Pol, another is the nuclear encoded RNA Pol. The first one is a eubacteria type multi-subunit. The first one is this chloroplast encoded. One thing is that from here. Chloroplast encoded RNA pol, that is the eubacteria type, and here multi-subunits present, and whose catalytic core, its catalytic core are encoded by chloroplast genome. Its catalytic core that is encoded by the chloroplast genome, and the core subunit, this core subunit that consists of alpha, beta, beta dash and omega. Okay, and these are encoded by the chloroplast DNA. So the core is encoded by the genome, and the core is made up of alpha, beta and beta dash and omega. Here, and alpha factors are encoded in the nuclear DNA, but in case of nuclear DNA, only the alpha factors that are present. And second one means is that nuclear. I think this is the first one, chloroplast, and second one is the nuclear. Nuclear encoded that RNA pol is the nuclear encoded T7 fast type and single subunit enzyme. So here in the second type there is only single subunit present, but in case of chloroplast, that is the nuclear chloroplast encoded RNA pol, that consists of multi subunits, but nuclear encoded RNA pol that consists of only single subunits. And here in the nuclear encoded RNA pol, there alpha factors has important role, but there are another factors also present that make the core region, and these are the alpha, beta, beta dash and omega. Clear? So this is the short explanation of the nuclear RNA polymerase, which is mainly responsible for the RNA transcription process. So in next video, I will try to explain about the promoter, eukaryotic promoter, which are responsible for the RNA transcription process, eukaryotic RNA transcription process. So continue with my channel. Thank you.